The rest of this session is going to be focused more on research, both internal and external to FDA. And I'm going to start off by talking about the multi-attribute method, which is one project that we're working on that's ETT inspired. So it's a nice kind of transition between ETT and our research. So the overview, I'll go over the learning objectives, kind of describe what MAM is, and talk about our research and the future of our research. So the learning objectives are to understand the basic workflow, what MAM is, understand the benefits and concerns that may be related to this method, and the 4 points to consider that we've developed regarding MAM. So, what is MAM? MAM is the multi-attribute method, and it is an LC-MS-based peptide mapping approach that's being proposed as a control method for therapeutic proteins. So it's been proposed to ETT by a few different groups as a replacement for conventional QC methods. And this has inspired us to do an OPQ lab-based research assessment. So on the right side of the screen here, you'll see kind of a basic overview. And this figure is taken from a commentary that actually just got accepted yesterday to analytical chemistry, and several things in the rest of this talk are going to be from that. So keep an eye out for that paper. It should be coming out pretty shortly. So basically what you have with the multi-attribute method, and this example is using a monoclonal antibody, but it's not necessarily limited to that. What we've seen at this point is mostly MABs and fusion proteins, though. So basically you do a quick digest, usually using trypsin. The time period of that is shorter than your general tryptic digest in order to move the process along and also limit digestion artifacts. So then you have a tube full of peptides, essentially, and you'll do LC-MS in order to do your method development. So in your method development run in the box on the upper right side, you'll be doing LC-MS, fully characterize your product through this method. So you'll look at all the different peptides that are in your digested sample, do a database search, and then determine what relevant PQAs or CQAs you would want to look at in the future. After you fully develop this method, you go back to just doing LC-MS. So you don't have quite as much data there. And then there are kind of two different major aspects to this method. So you'll have the targeted attribute quantitation and the new peak detection. So for the targeted attribute quantitation, what you'll be doing is monitoring the relative abundance of various modifications generally, or truncations, things like that, that are the PQAs that you determined in your method development. The other step of this is the new peak detection. And in the new peak detection, you'll be using a differential algorithm to compare your test sample to a reference sample to see if any peaks have changed. So this is a slide that people from industry generally like to show, which is looking at the properties of MAM and what people think it might be able to replace. So the basic idea here is that there are a lot of different characteristics that you should be able to assess using MAM that cover several different conventional methods. And I'll go into more specifics of this in a few slides. So the general benefits of MAM are that you're testing multiple attributes at once. You're not just looking at all of the acidic species, you're looking at specific modifications that make up that larger subset of species. So this allows you to have fewer instruments and fewer assays. It also gives you more detailed information at the molecular level. So because you're analyzing site-specific modifications, it will hopefully allow you to have tighter control over your product. It also allows you to differentiate between species that may overlap using chromatographic approaches. So as I was saying, if you have a big peak and some sort of chromatographic approach, any of them, might be multiple species in that peak. Because you're looking at the site-specific modifications with MAM, you should be able to differentiate between those because you're using the mass based on the mass spectrometer. And then finally, for the general benefits, we have the new peak detection feature, which should allow for control of unexpected new modifications. So something that you're not necessarily looking for should hopefully appear when you're doing the new peak detection analysis. So based on both our internal research as well as publications and presentations we've seen at conferences, as well as information we've received through the ETT, we've come up with 4 major points to consider. And I'll go through each of these individually and then talk a bit about how our research relates to these. So for this, we have the 4 major points that we've come up with, and these are, again, outlined in that commentary I mentioned earlier that should be available soon. So the 4 major points that we've come up with are risk assessment, method validation, the new peak detection feature, and the comparison to conventional methods. So for a risk assessment, one thing that we found about MAM is that it's definitely product and CQA specific. So it's not going to apply to just absolutely everything. It's going to be very specific, and you'll want to make sure you weigh the benefits and risks for implementation of this approach. You don't want to just throw it on everything. So as I said, this is product and CQA specific. You want to really think about how this is affecting your process. One potential risk example that I showed on the bottom here is a loss of information about clipped species. So if you're doing a triptych digest, you wouldn't necessarily be able to tell the difference between the 2 species on the left because a long peptide and 2 separate peptides, if it cleaves at a triptych cleavage site, would result in the same peptides in the long run, so you'd be analyzing the same thing. Whether or not this is important is going to be specific to your product. So a risk analysis or risk assessment around this type of thing would be useful in order to figure out whether it's appropriate or not to use MAM here. For method validation, as an analytical method, MAM needs to be validated. However, there has not been much use of mass spectrometry for proteins in QC up to this point, so that people don't have a lot of experience validating mass spec-based methods. What we're saying is that you can base your validation on ICH guidelines as well as FDA guidances, and I've listed some relevant guidance documents in the box. What we've found from looking at it is that some of the more challenging aspects to validating an MAM method are precision, LOD, LOQ, and system suitability, and looking at how you really go into that for this sort of approach. So for new peak detection, so this is one of the new features of this method that can also be a little hard to validate, and so we're interested in figuring that out as well. But so it allows for detection of changes that aren't directly monitored by your list of PQAs that you've already characterized, and it should be able to detect unknown impurities, especially if you're using it in a stability setting. What we've found with new peak detection is that it's really highly dependent on software parameters. So what software you're using for this comparison is critical, as well as certain parameters that you put into the software. So what retention time window you're using, what mass accuracy window, and what the peak detection threshold is. So my example in the picture here is looking at the peak detection threshold. So if you have an LCMS chromatogram of your Reproducibility standard, as you see in blue, and one of the test sample, which you see in orange, if you do a differential analysis, you can see that there are several peaks that are present in one or the other. And so depending on where you set your detection threshold, you may or may not see some of these peaks, which if you set your detection threshold too low, you might see a lot of noise peaks that aren't really real changes. If you set it too high, you might miss real changes. So it's really critical to look at your instrument and at your product and work on your workflow to determine specifically for you what that detection threshold is. All right, so our fourth point to consider is conventional method comparisons. So this is going to loop back to our risk assessment. So how you compare these is going to be dependent on that. And it'll help you understand the advantages and disadvantages of using this method. And you would want to perform these comparisons during method and product development. However, because you're not comparing apples to apples, you're comparing apples to oranges, your measurements may not correlate. So in the table here, we can see what the conventional method is measuring and what MAM is targeting. So the most straightforward comparison is looking at your glycosylation profile. So the conventional method here would be to acylic glycan profiling to look at the N-glycan distribution. But with MAM, you're looking at glycopeptides. And that's actually a pretty straightforward comparison and should be fairly similar. However, other methods that are being proposed to be replaced by MAM, including charge variant analysis by CEX and clip species analysis and other size and purity and variant analysis using RCE SDS, it's a little different what you're looking at. So for CEX, you're looking at charge variants, but with MAM, you're looking at specific post-translational modification, specific NNC terminal variants, and sialylated species. So how you can compare those is really important in determining whether this method is appropriate for you. Additionally, with the specific clipped species, theoretically, you might miss things as compared to the RCE SDS. However, hopefully, with the new peak detection feature, you should be able to catch those differences if you're not directly monitoring them anyway. A few additional considerations that we've kind of come up with that don't quite fall into the buckets, but really can probably be looped into the risk assessment, are that you might lose information at the protein level. So in the example shown on the right, we have a few different distributions of a unspecified modification across different particular antibodies. So if you generally have 2 modifications, one on each chain in half of your molecules and on neither chain in half of your molecules, you wouldn't be able to necessarily tell the difference between that and having all of your molecules with 1 modification on each chain. And whether that's important or not comes back to the risk assessment and looking at your specific product and whether that affects the product quality. So again, because you're digesting the protein, you're losing you may be losing some information about the protein as a whole. But again, it depends on whether that's important depends on if that affects the safety or efficacy of your product, and it's going to be case by case based on a risk assessment. And finally, you just want to make sure that this method is fit for your particular purpose. You want to demonstrate that your new QC method is monitoring everything that it needs to monitor, and that you're not losing information when you're switching to this method as compared to your conventional methods. Okay, now switching gears into our research at FDA. So based on some of the information we got from ETT, we established an in-house MAM platform in order to better understand MAM ourselves in our own hands. So for our research, we've been using rituximab as a model protein, mostly because it's pretty easy for us to get a hold of. And we also have some unapproved product in addition to the approved product, so we can do some comparisons. What we focused on and what I'll talk some about today are method validation, so particularly system suitability, precision, and LOD/LOQ. New peak detection. Today I'll be focusing on the force degradation, though we've also been doing some user-to-user comparisons. And then the conventional method comparisons focused again on force degradation. So as a method overview, here's a diagram of rituximab and the particular PQAs that we've been assessing. So we looked at 21 different product quality attributes across 11 different sites on the protein. And we were able to distinguish using our MAM platform between our approved and unapproved products for 10 of those PQAs. So first off, with method validation, I want to mention system suitability, which is really important for this method. We specifically here are using a Pierce peptide mix, which contains 15 isotopically labeled peptides. We focused on using 12 of those for our system suitability testing because they were more reliable in our particular instrument. And using those and historical data over about a year's time, we set relative abundance and retention time limits based on our internal data for those particular peptides. We also set limits for the variability, so the percent CV of the retention time and relative abundance, in order to make sure that things aren't changing too much and our instrument is actually suitable for this use. We've also set guidelines or baselines for our mass accuracy, resolution, and signal-to-noise ratio. We have been using this Pierce peptide mix, but it may not be suitable for everyone. You might want to try using something a little more similar to your particular product in order to make sure that your instrument is performing as well as possible. I know some people have been looking into using NIST-MAP as a standard, and other people have been using just looking at using a reference sample of their own product. And that's up to you, and if you provide a good justification, then we can talk about that. So we've also looked at reproducibility and precision. We had 3 users who did 3 different digests and 3 injections each to do this initial study. And we found that generally our results were reproducible across users and digests, but we did find pretty high variability specifically for low abundance oxidation sites. So one thing that we found is, and this is pretty well known already, is that oxidation is really liable to artifacts, particularly in the digest. So it's a little more variability. What we did find is that variability and changes in the levels of oxidation were really apparent based on the experience of the user. So we had 3 different users with varying levels of experience doing this method, and the one with the least experience had the highest levels of oxidation, and that skewed our variability. So what we took away from that is that you should really make sure that everyone is really well trained, you have SOPs in place to make sure that your data is consistent, and that you should be monitoring your data over time to really ensure that the people using these methods are capable. This is a little more about our user variation in this, looking specifically at the oxidation. And user C here, with the highest levels of oxidation in all these sites, is again the least experienced of the 3. So we definitely saw there was more variation when we had less experience. So for new peak detection, we looked at a forced degradation study. So we had a 28-day forced degradation study at 40 C and 75% relative humidity. And when we looked at our PQAs over time, we saw linear increases in both oxidation and deamidation. And when we looked at the new peak detection for this study, we found 1 new peak, and that was a change from an aspartic acid to an isoaspartic acid. And you can see it in this chromatogram here. There's this tiny little peak here. So we found that when we compared our 28-day sample to our day 0 sample. And when we looked at that particular peak over time, we saw that it had increased linearly over the time course as well. So that was a nice thing to find with the new peak detection. We found something, it was real, we could look at it over time. So using the same sample set, we looked at forced degradation, or the same forced degradation sample set, we looked at method comparisons. And here we compared the MAM of one particular deamidation site to our acidic variants from our charge variant analysis. And we saw that they both increased over time, but not exactly at the same slope. If we do a correlation, we see that it's pretty good, but it's not perfect. So what we'd want to do here is kind of go back and characterize our acidic peak and see what else is in there, and if we can find other MAM attributes that we can look at through MAM and see if those make it a little more linear. But you're not necessarily always going to be able to do that. So for a summary of this, there are 4 major points to consider that we've come up with, and that's the risk assessment, which you should be considering when you're developing MAM. Method validation, which we use method validation. We established our system suitability approach. We assess precision, reproducibility, LOD, LOQ, and more. Although I don't have time to go into all of those data today. For the new peak detection, we in-house have established suggested parameters for new peak detection and have used it to test forced degradation samples. And we've also done some method comparisons with our forced degradation. We've also done some glycan analysis as well, but it's a little less exciting, so I cut it out for today. Our ongoing and future research, we're currently running and analyzing data from a year-long stability study and accelerated stability studies to compare MAM and conventional methods over a long period of time and to see how well they compare and what we can do with that. We're also looking at a software comparison because, as I mentioned earlier, specifically for the new peak detection, it's really important what software you use and what it can do and how well it performs is really important. And we're also working on a site-to-site comparison between our Maryland and St. Louis locations. All right. I'd like to acknowledge a whole bunch of people who worked on this project, both in the Office of Testing and Research and the Office of Biotechnology Products and the Emerging Technology team. So, challenge questions. What does MAM stand for? Is it multi-attribute monitoring, multi-attribute method, multi-analysis method, or multi-assessment monitoring? Yes. yeah, B is correct. I threw in A just kind of to trick people because there is another method that's very similar that people are proposing that takes out the new peak detection portion and just focuses on the monitoring. And that can use a lower resolution instrument. True or false? Risk assessments for MAM should be product and method specific. True. Okay. And then finally, which of the following is not one of FDA's points to consider for MAM? Risk assessment, new peak detection, software comparisons, method validation, and method comparisons. C, correct. Good job, guys. Software comparison is important, but it's not in our official list of points to consider. Okay, and I think that's all I have. Thank you.